everyone. Myself Tejaswini Yadav from microbiology department of Dapoli Urban Bank Senior Science College, Dapoli. Here today we can study the phagocytosis. Phagocytosis is a non-specific defense mechanism of the body. In 1883, scientist recognized that the phagocytosis is protect the body from the microbial infection. Here we can study first the definition of phagocytosis. What is the phagocytosis? The phagocytosis is a process in which the recognized ingestion and digestion of a extracellular microbiological matter is called as the phagocytosis. This phagocytosis process is completed into the 5 steps. That is, first step is a chemotaxis, second step is a attachment, third step is a ingestion, fourth step is a digestion and killing of microbial cell, and last is a exocytosis. Here we can first study the chemotaxis. Chemotaxis is a first step in the phagocytosis process. In chemotaxis process, the phagocytic cells which are present into the human body are moved towards the microbial cell because of the phagocytic factors. These phagocytic factors are synthesized by the microorganisms which are entered into the human body. This is the chemotaxis. Then second step is a attachment. After the accumulation of phagocytic cells around the microorganisms, they are attached to the phagocytic cells, and the attachment of phagocytic cells and the bacterial or microbial cells are carried out in the process of attachment. This attachment is depend on the two mechanisms. That is, first mechanism is the opsonin dependent mechanism. Second mechanism is a opsonin independent mechanism. Here we can first study the opsonin dependent mechanism. First we can study what is the opsonin. Opsonin is a serum component like that of antibodies. Next, this opsonin is held to the attachment of microorganism to the phagocytic cells. Like that, the opsonin is formation of bridge between the phagocytic cells and the microorganisms. This is the opsonin dependent mechanism. And then second is the opsonin independent mechanism. In opsonin independent mechanism, opsonin does not require to the attachment of microorganisms to the phagocytic cells. Okay? Because of the phagocytic cells contains specific as well as the non-specific receptors. These receptors recognize the surface structure of the microorganism and they bind with that microorganism. Here, this is the opsonin independent mechanism. Here, the attachment step is completed. After the attachment, the ingestion process is carried out. In the ingestion process, after the attachment of microorganism to the phagocytic cells, the phagocytic cells are produced or formation of a seropodia. And after the formation of seropodia, the microorganism is entered into the phagocytic cells. Then to form a vacuole around the microorganism and to formation of a phagosome. Also, in the phagocytic cells, nucleus, Golgi bodies are present. These Golgi bodies produces a different enzymes and proteins like a lysozyme. This lysozyme enzyme is attached to the phagosome and to formation of a phagolysosome. This is the process of ingestion. After the ingestion, the digestion and killing process is carried out. Here, the fourth step is digestion the consumption of oxygen rate of phagocytic cells is increasing, and this process is called as the respiratory burst. Then what is respiratory burst? The consumption of oxygen rate is increased is called as the respiratory burst. This respiratory burst is effect on the hexose monophosphate pathway, and the activity of this hexose monophosphate pathway is increased. During the activation of hexose monophosphate pathway, the NADH is released in the higher concentration, and this NADH is oxidized and to formation of a NADH. During the oxidation of NADH, different toxic products are synthesized. For example, singlet oxygen and hydrogen peroxide. This singlet oxygen or hydrogen peroxide is effect on the microorganism, and this singlet oxygen or hydrogen peroxide is toxic to that microorganisms, and they are carried out the digestion of microorganisms. This is the oxygen dependent mechanism which are carried out in the digestion of the microorganism. Then second is a oxygen independent digestion or the killing of microorganism. In oxygen independent mechanism, the oxygen cannot be required for the digestion of microorganisms. In this mechanism, the lysozyme which is present into the phagocytic cell act on the microorganism, and they digest the microorganism. This is the digestion and killing process of the microorganism. And last is exocytosis. In the exocytosis process, after the completion of digestion, the digested matter of the microorganism is absorbed in the cytoplasm of phagocytic cells, and the undigested particulate matter of the microorganism is thrown out through the phagocytic cells with the help of exocytosis process. And this exocytosis process is effect on this exocytosis process is exactly opposite to the ingestion. Here it is the phagocytosis. Here we can explain the phagocytosis process diagrammatically. Here this is the phagocytic cell. This is the phagocytic cells. Here are different microorganisms which is also called as antigens. These antigens are attached to the macrophage. After the attachment of antigen to the macrophage or the phagocytic cells form a pseudopodia. Here these are the pseudopodia. With the help of this pseudopodia, the antigen is entered into the phagocytic cell. After the entry of phagocytic cells, the vacuole is formed around the antigen or microorganism, and is called as the phagosome. After the formation of phagosome, the phagolysosome is formed. Here that is the phagosome. Phagosome means the vacuole which is present around the microorganism or antigen. Here lysozyme enzyme is attached to that phagosome, and hence is called as the phagolysosome. metabolization and digestion of microorganisms. This is called as the phagosynthesis. This phagosynthesis is completed into the 5 steps, that is chemotaxis, attachment, ingestion, digestion and exocytosis. This phagosynthesis